Good afternoon. Good afternoon. Good afternoon. I'm Rick Myers, President and Science Director of the HudsonAlpha Institute for Biotechnology. I'm sorry we can't meet in person now, but this technology will allow us to tell you about our updates on Alzheimer's disease. Even though COVID is occupying all of our minds now, so does Alzheimer's and other neurodegenerative diseases and all the other research that we do here at HudsonAlpha, and we're welcome the opportunity to tell you about that today. In a moment, I will introduce our speakers, but first I would like to thank Hightower Twickenham for sponsoring this event today. In particular, we'd thank Wes Clayton and his colleagues for all the support they've given us over the years, and especially in our neurodegenerative disease research. I'd like to introduce you now to Rob Warren.
Thank you, Dr. Myers, and thank you to the rest of the team at HudsonAlpha as well. On behalf of Hightower Twickenham, we are incredibly proud to be able to partner with a wonderful organization like HudsonAlpha to be able to bring programs like these to our community. Over the last several months, as the entire world's focus has been elsewhere, there are still millions of individuals and millions of families who continue to struggle with the terrible effects of Alzheimer's disease. As our focus has been elsewhere, we take great comfort in knowing that the scientists at HudsonAlpha continue to work towards breakthrough technologies and discoveries for early diagnostics and effective treatments for a terrible disease. It's those scientists that give us comfort and hope in knowing that we can live in a world without Alzheimer's disease. So thank you. Thank you, Dr. Myers. Thank you to your team. Thank you to your scientists. And thank you for joining us for a wonderful event today in a new format. Welcome to the 2020 Alzheimer Update.
Thanks, Rob. We have three speakers today to tell you about our research on Alzheimer's disease. And our means HudsonAlpha Institute for Biotechnology, which is a nonprofit research and teaching institute here in Huntsville, but also we're going to hear from one of our collaborators from California. Alzheimer's disease is a terrible scourge, as we all know. It affects our lives in so many different ways. And just thinking about the numbers is how you to then start treating the disease much earlier than otherwise. So one way to think about this is that you draw blood from someone. It's not very much. It's just a couple of tablespoons of blood at the most. And then you do a centrifugation step to actually separate the cells in the blood from the liquid in the blood. And that liquid that we want to go after is called plasma. It's clear stuff or slightly yellow stuff at the top. So by doing that, we are now taking that and then looking for the presence in the plasma of something that might be indicative of Alzheimer's disease. And that can be proteins, it could be DNA, it could be RNA, which is DNA's cousin, it could be lipids, which are fats, and it could even be small molecules called metabolites from breaking down of various things in our cells. So people have been looking for such, we call these biomarkers, people have been looking for these biomarkers for a long time. And we, because we're experts at HudsonAlpha on looking at DNA and RNA especially, are spending our efforts on those for Alzheimer's disease. And in particular, we are developing tests where you can take a little bit of plasma and from about 150 people with Alzheimer's disease and about 150 without, and see if we can find something different in the DNA or RNA that's floating around in the plasma. We're making a lot of progress in this. It's an exciting project. I hope we will have answers to this within the next year, but we're working hard on it to make this happen. So here's a mock-up of what we would like to be able to see in a blood test for Alzheimer's disease. You see an image like the one here where you've looked at a bunch of people without disease and you get something like that. And then you look at a bunch of people who have the disease and you do measurements and you get a different set of images. And those images then are what we call biomarkers that could be the blood test for Alzheimer's disease. So that's one of our projects that we're doing here at HudsonAlpha. Let me now introduce you to Dr. Nick Cochran, who will tell you about another one.
Thank you, Rick, and good afternoon, everyone. Thank you for joining us in this unusual way, but it's very important that we continue this important work. These neurodegenerative diseases don't stop because of this pandemic, and so it's important that we continue our work in whatever way that we can, and that's what we've been doing. So I'd like today to talk about a problem that Rick has been working on in his lab for a long time that now we're applying to these diseases, and that's the problem of gene regulation, or how genes are turned on and off, and then how we can use that knowledge to work towards potentially new treatments for neurodegenerative disease. And for that, I'd like to first back up and give a little bit of introduction on what gene that we're going to be talking about and why it's important. So there are two main proteins that build up in the brain in Alzheimer's disease, and they're shown here on this slide. So in a healthy brain, ideally this slide would be blank. There wouldn't be any of these aggregates that are shown. And you've probably heard of amyloid beta plaques, so that's these aggregates that build up outside of cells in the brain. And then you may have also heard of tangles of tau protein, and those build up inside neurons in the brain. And tau is the gene and the protein that I'm going to be talking about over the next few minutes. And the reason I'm talking about it is because it's very important for permitting the disease to run its course. So you need both amyloid beta and tau for Alzheimer's disease. And tau, importantly, builds up over our entire lifetimes. So it's always there in the background, building up over time. And this is a plot that shows that. You don't need to look at the specifics of the bars here. What you need to see is just this blue getting darker with increasing age. And what that indicates is that tau is building up more and more over time. And that process permits for things like Alzheimer's disease to occur. Now, the field has come to learn that we may be able to just reduce tau levels. So tau is probably important during early development, for example in children. However, once we get to be adults, it's likely that we can reduce tau levels without many harmful effects. We see this for sure in animal models. And so these are animals that are engineered to develop something that's sort of like Alzheimer's disease. And when we reduce tau in those animal models, there are beneficial effects. There are also a lot of drug companies trying to reduce tau because of this evidence that we've generated as a field that lowering its levels may be beneficial. But a lot of those techniques are still doing things like spinal taps to be able to deliver the machinery that you need into the brain to reduce levels of tau. So, as I mentioned, Rick's labs for decades has been working on gene regulation, or how genes are turned on and off. So we could apply this knowledge to tau and find ways potentially to reduce its level, which may have benefit. So how can we find these on-off switches? This is a schematic of DNA and then how it's organized. So DNA is packed up by lots of machinery into tight bundles that then make up our chromosomes and our cells. And understanding the process of how this occurs and how it's opened up to allow for genes to be expressed can allow us to know then how genes are turned on and off. To give a little more detail, if we zoomed in on one of those strands of DNA, there's DNA that then is in front of, for example, a gene like tau. And this would be the main on switch for tau. And what we see is that DNA is organized into these loops, and there are actually many other types of on switches that are nearby to the main on switch for tau. So one way to understand how a gene like tau is turned on is to find these on switches, and we have tools in the lab to do that. The other way to understand how a gene like tau is turned on is to say, okay, what's flipping that switch? What are the hands that are coming in and flipping that switch to turn on tau? And that is a group of proteins that are called transcription factors. Here I've just arbitrarily labeled them A, B, C, and D. We don't know what they are. That's part of the goal of the research, is to discover what they are. And it's really important to understand what they are because proteins are what can be targeted with, for example, a drug that may take the form of a pill. So for example, a protein may have a pocket that a drug could sit in and inhibit its activity. Or perhaps even you could find a molecule that disrupts the interaction between two proteins, and so it keeps these on switches from being able to work together to turn on tau. So now I've shared the general idea of what we want to do with you, but then the question is, where are we with this work? How much progress have we made? And the exciting news is that we've made quite a bit of progress so far. So with initial support from the Memory and Mobility Program, we were able to get some preliminary data that allowed us to obtain additional funding from the Bright Focus Foundation, and that's a grant that's currently funded that we're currently addressing this problem. And then with that data that we were able to generate, we've now applied for an even larger grant from the National Institutes of Health that will allow us to continue this work and add to the evidence for this idea and keep working towards that idea of understanding how tau is turned on, which of course we hope will lead to efforts to target tau therapeutically. Now, I put up this about a presentation that we made at a meeting sponsored by the Alzheimer's Association and talk about a recent award that we won for presenting this work at this meeting. Not to brag about myself or my ability to present this work, it's that the work speaks for itself and that the field recognizes that this is an important way to address this issue. So out of over 100 abstracts, this was one of 20 chosen to be presented as a platform presentation in front of an entire audience at an international meeting. And of those 20 presentations, this was one of 4 that was selected as one of the best presentations. And so we're really excited about that and excited that the field is excited about what we're doing here. So with that, I have been so excited to be with you this afternoon and share a little bit about what we're doing and applying our expertise in gene regulation to this very important problem in neurodegeneration. And now I'd like to hand it over to Dr. Jennifer Yokoyama. She is an assistant professor at the University of California, San Francisco. And she has been a longtime collaborator with us here at Hudson Alpha. We work on several projects together. And today she's going to tell you some about what type of work she does in her lab and also about some of the ongoing and future collaborations that we're working on together. Thank you very much.
Thank you, Dr. Myers and Dr. Cochran, for the nice introduction. And thank you to all of you viewers. Welcome to my living room. I'm happy to tell you today about some of the work that we do in my neurogenetics and aging lab at the University of California, San Francisco Memory and Aging Center. So our lab is really interested in how genetic variation contributes to brain structure and function, both in healthy aging as well as in neurodegenerative disease. And so we study the relationships between genetic variation, which you learned about earlier, along with structural changes of the brain measured by neuroimaging or MRI, and then correlate those with changes in clinical and cognitive function. And so as Dr. Myers said, there are many different ways that studying genetics of diseases like Alzheimer's disease can really assist our understanding of the causes of Alzheimer's disease, can help us with early identification or detection of disease risk, and also lead to treatments. And so by comparing differences in patients versus cognitively healthy older adults, we can do all of these things. And today I'm going to tell you just a bit about some of the work we've been doing to try to work on early detection of disease risk. So we have successfully combined information from multiple different markers across the genome to make predictions about each individual's risk for Alzheimer's disease. And so you can see in this graph, this is a survival analysis. And what we see is that people at the top are really high up there, and they don't really convert to Alzheimer's disease. So they have very low risk in blue. On the other hand, compared to average risk, there are some individuals who carry a lot of genetic risk factors for Alzheimer's disease and show a much higher risk than other individuals. And so while these tools are very helpful, there are some limitations to these types of measures as well. And among others, one of the most important limitations to consider is that they are very dependent upon the populations or people they were developed in. So what do I mean by that? So let's take a simple example. Let's say we want to try to create some sort of very simple measure that we can apply to assess the ripeness of a banana. Okay, stick with me here. So there are different ways to try to predict banana ripeness. And so some ways might include the density or texture of the banana. Is it soft? Is it firm? Other aspects which we often use are related to color. So we know that a green banana is not very ripe and a brown banana is way too ripe. So we can combine this type of information about features or measures that you can take on every type of banana and create a banana ripeness score, which we would be in parallel thinking of as a genetic risk score, for example. So in this case, we may consider, okay, well, let's see. We go on a spectrum from a firm banana to a very squishy banana. And also we have this other measure of color, where green bananas are often very firm and brown bananas are pretty squishy usually. And so in this way, we can assess low ripeness or high ripeness on this nice spectrum. And then we can apply it to many different bananas and say, oh, okay, I see where you lay on this scale and I can assess the level of ripeness that this banana has. Okay, so that makes sense for bananas, but how generalizable is this? So if you apply these same measures of texture or density and color to related banana species like mini bananas or plantains, then they would probably work pretty well. But what happens if you try to apply them to a grapefruit or an apple? Then it's possible that some of the measures are not quite right anymore, and they won't make as good of predictions about fruit ripeness compared to the measures as applied to other bananas. So in this fruit analogy, we would basically say, okay, well, we really need fruit family specific ripeness scores. And maybe they all share in common this general trend that firm fruit are not very ripe and squishy fruit are really too ripe. But there may be subtle differences in the color gradient that corresponds to that firm versus squishy or not ripe versus too ripe measure. And so in this way, if we have a banana specific, citrus specific, or apple specific measure, we can account for differences in each of those groups while still being able to measure the same thing, ripeness. And so this is something that we are thinking about now in the context of human beings. So researchers in human genetics know that by studying diverse genetic populations, and what I mean by that are individuals of all different genetic or ancestral backgrounds, can really help us learn more about diseases like Alzheimer's. And so by analyzing these more diverse populations or groups of people, we can learn a lot more, not only about the genes that contribute to Alzheimer's disease risk, but also learn information about how each different type of variation may contribute subtly in different ways to disease risk across each population. And so we not only are interested in diverse populations because of this genetic richness, but also because these populations are often the most affected and in threat of problems due to social inequities and health disparities. So we know that social determinants of health are really rooted in socioeconomic inequities, driving ill health and dementia. And this is seen now very clearly, even in the context of the COVID-19 pandemic. So this is something that is really important and something we would really like to address. In addition, we know that dementia due to diseases like Alzheimer's disease will rise disproportionately high in lower and middle income countries compared to high income countries. That's what you see here in this graph, where by the year 2050, we anticipate or estimate that there will be a much greater increase in the millions of people with dementia in lower and middle income countries. And finally, in many cultures, including the United States, Stigma of dementia augments socioeconomic inequity and adds to the stigma that's already present in aging. And so there is here this triple threat of health inequities that we are really hoping to address through this following study that I'm going to tell you about. And really, the goal of the study that I'm going to tell you about is to make sure that we have genetic information that's relevant to every population in order to make the best predictions of disease risk that we can for each person. So this very busy slide is basically to show you the huge number of people that are experts in neurodegenerative disease and dementia across the country, and particularly in South America, in Latin America. And they include our colleagues, Dr. Rick Myers and Dr. Nick Cochran at HudsonAlpha. And our goal of this project, which is just started this year, is really to assess correlations or relationships between genes and brain and behavior across different patients with Alzheimer's disease and another disease called frontotemporal dementia across diverse populations, really anchored in South and Latin America. So you can see here the list of the different countries currently involved in this project. And I can tell you that every day I think this list continues to grow. And also note just a very quick schematic of all of the different types of studies that we're planning to do with this large group of people. And we hope and we plan to collect information from patients and healthy controls from each region, numbering over 4,000 total participants, and all of them will be genetically characterized at HudsonAlpha. So with that, I would just like to end by acknowledging the funding support for our research, including that collaborative project I told you about, as well as for my lab and the members of my team. You're welcome to learn more about the research that we do in genetics and neuroimaging at our website. And I'd just like to thank you again for taking the time to learn a little bit more about our work. Take care.
Thank you, Nick and Jennifer. Boy, is that exciting research. And I will say, we don't do any of our research in a vacuum, and it has just been wonderful to be collaborators with Jen and her colleagues at UC San Francisco and at other institutions as well. I'd also like to thank all of those of you who have supported our Memory and Mobility Fund. This work wouldn't be possible if we didn't have funding, and the M&M Fund, as we call it, has made a huge difference in our ability to do these projects and several others having to do with Alzheimer's disease and other neurodegenerative disease. Someone who has supported our research in neurodegenerative diseases is Carol Tivapal. Carol and her family have been directly affected by Alzheimer's disease, and this really comes home to them. Carol, would you like to say a few words, please?
Thank you, Rick. Hello, I'm Carol Tivapal. My husband Jim and I have been supporting HudsonAlpha's Memory and Mobility Unit for several years. We've got a very personal interest in HudsonAlpha's Alzheimer's and other neurodegenerative research. By the time my dad passed 9 years ago, he was starting to exhibit some signs of dementia. And within a few months of his passing, my mom sort of flipped into dementia. That's strong emotional events can trigger that. And she's been in a long-term care facility for memory and mobility since 2011. Jim's mom started to exhibit some vascular dementia prior to her passing in 2000. So I'm sure you can understand that we truly have a vested interest in understanding this family of terrible, devastating diseases. Prior to finding out about HudsonAlpha's focused program to try and understand and find ways to defeat these horrible neurodegenerative diseases, we supported other Alzheimer's related programs trying to understand. But after finding out about HudsonAlpha's Memory and Mobility Program to support the genomics research of neurological diseases, we retargeted our resources and are focusing on HudsonAlpha. HudsonAlpha's research works to pinpoint genetic causes of Alzheimer's disease and dementia. If we can understand the genetic causes of neurodegenerative diseases, It should enable researchers to better identify promising treatments and hopefully a cure. So I follow their research, I attend their lectures, and I watch their blogs because Hudson Alpha gives me hope. I really believe in the work that they're doing at Hudson Alpha, and I'm excited about the discoveries that they're making. For your loved ones, and maybe even yourselves, I encourage you to join us in supporting Hudson Alpha. They're working the big problems to arrive at the great solutions. Please consider joining us and others in making a gift to Hudson Alpha. A gift can make a difference in the fight against Alzheimer's and other neurodegenerative diseases. We will all thank you.
Thank you, Carol, for those words, and thank you so much for your support of the Memory and Mobility Fund. We hope all of you will consider donating to the Memory and Mobility Fund. This work is really important. We've accomplished a lot because of the M&M Fund, and we have a lot more left to do. Thank you for joining this virtual Alzheimer's disease update. We will keep you posted on our progress and hope to hear from you again soon. Please be safe, and thank you all for coming.